Hello everybody. Today's topic of discussion is lipid metabolism. This is the class of continuation of previous classes. That is the our topic of lecture is chapter is lipid metabolism. So how the LDLs get metabolized after delivery of cholesterol ester from liver to extrahepatic tissue. So LDL, the full form of LDL is low density lipoprotein. The main constituents of LDL, that is the protein content, is ApoB100, that is present in the surface, free cholesterol and phospholipid, and also the ApoE, and in the center main lipid is cholesterol ester. So after delivery of cholesterol ester to the skeletal muscle and adipose tissue, it is targeted to liver. So how the liver recognize the LDL? Recognize the LDL. In the LDL, there is a ApoB100. In the liver, there is a LDL receptor on the cell surface, and this LDL receptor recognizes the ApoB100 molecule as well as ApoE molecule. So ApoB100 molecule that is present in LDL is a single. It is composed of single polypeptide chain made up of 4,500 amino acid, and it has got LDL receptor recognition site. It is a product of ApoB gene, and in the hepatocyte it is fully translated. The ApoB gene is transcripted to ApoB mRNA, and it is fully translated, 100% translated, 100% length of ApoB mRNA is translated to form ApoB100. So the ApoB100 contains LDL receptor recognition site. When the LDL molecule reaches the surface of near the surface of the hepatocytes, the LDL receptor recognize the ApoB100 molecule and get attached and forms the coated pit. And coated pit is lined by a particular protein. It's called clartrin. After that, the coated pit is formed a vesicle. Vesicle means a rounded structure. Inside it, the LDL molecule remain attached with the LDL receptor with ApoB100, and the coated vesicles is lined by clartrin molecule. and this is called endosome. Then lysosome come to act and joins with the endosome to form endolysosome. As we all know, the lysosome contains all the hydrolytic enzymes and it degrades, hydrolyze all the structure of LDL molecule except LDL receptor, and so the LDL receptor recycle back to the and transported to the hepatocyte cell surface by the recycled vesicles. By the action of hydrolytic enzyme of the lysosome, all the protein of LDL gets hydrolyzed to form amino acid, and this amino acid is either utilized for the formation of protein or broken down into its catabolic end product. Cholesterol ester is hydrolyzed to form fatty acid and the cholesterol, and fatty acid is reutilized for the esterification or catabolized into production of ATP. And ultimately, what will be the fate of cholesterol? Cholesterol, the main fate is within the hepatocyte is bile acid synthesis. Second is it forms the cell membrane. As we know that the one of the constituents of cell membrane is cholesterol. Another fate of the cholesterol is re-esterification of cholesterol to form cholesterol ester with the enzyme called ACAT, that is the acyl cholesterol acyl transferase. Next fate is cholesterol inhibit the HMG-CoA reductase enzyme, that is key enzyme of cholesterol synthesis. So ultimately cholesterol biosynthesis will rate of cholesterol biosynthesis decreases, and last function of the cholesterol is it downregulate the LDL receptor synthesis at the genetic level. So when the cholesterol level within the hepatocyte is more, there is a decrease number of LDL receptor on the hepatocyte. So cholesterol entry within the hepatocyte will decrease. So these are the fate of cholesterol. Then you have to know the structure of LDL receptor. Why? Because how it recognizes the ApoB100 and why the only LDL molecule, why the ApoB100 molecule is recognized by the LDL receptor, not the ApoE molecule. So LDL receptor is a glycoprotein molecule. The protein part is composed of 839 amino acid and it has got 5 domain. What is domain? Domain means it is a particular part, it is a part of polypeptide chain, has got definite function, individual function. So from N terminal to C terminal there is a 5 domain. First domain is ligand binding domain. It has got the 7 spiral part, and in this part there is a ApoB100 recognition site. In ApoB100 molecule, there is a positively charged amino acid rich in positively charged amino acid, and in the LDL ligand binding site there is a negatively charged amino acids. So ApoB100 molecule rich in positively charged amino acid, and the LDL binding domain of LDL receptor rich in negatively charged amino acid. So there is a electrostatic interaction between these two. So the LDL molecule get attached with the ApoB100 molecule, and in this way ApoE molecule also get attached with the LDL receptor. ApoE containing molecules lipoprotein can attach with the LDL receptor. So the receptor is called ApoB100E receptor. LDL receptor is also called ApoBE receptor. But due to the EGF domain, that is the next EGF domain that contains rich in negatively charged amino acid, and the function of this EGF domain is that it serves to maintain the correct orientation of the ligand binding domain on the cell surface. The specific orientation determined by this EGF domain is required for the binding of LDL to the receptor, but not the binding of ApoE containing lipoprotein. So the EGF domain is helpful. Is it responsible for the only ApoB100 containing lipoprotein binding with the LDL receptor. How the LDL receptor dissociate the LDL molecule in the endolysosome. We have discussed that the LDL receptor can bind with the ApoB100 by electrostatic interaction. But how the LDL molecule gets dissociate from the LDL receptor in the endolysosome? There is a protonation of the negatively charged amino acid of the LDL receptor occurs in the acidic lysosome. So the LDL receptor dissociate the LDL molecule, release the LDL molecule from the LDL receptor. In a acidic endolysosome, there is a protonation of the negatively charged amino acid of the LDL receptor occurs that leads to release of LDL molecule. So the LDL molecule get detached from the LDL receptor. Next domain is rich in serine and threonine residue. Here the oligosaccharide residue chains are attached. So the whole molecule, that is the LDL receptor molecule, is glycoprotein. It's called glycoprotein in nature. Next, the domain is membrane spanning domain, that is rich in hydrophobic amino acid. That means it passes through the cell membrane. And last part domain is cytoplasmic domain, that is remain within the cytoplasm. It is important to targeting the LDL receptor to the coated pit region. So that's all. In this way, this is the part of LDL receptor, and in this way LDL molecule get catabolized within the hepatocytes. We have discussed these two parts, that is the LDL catabolism. LDL is catabolized in this way, as well as LDL receptor structure. And that's all for today.